Chapter 6, Interactions Between Cells. I brought my own batteries tonight. Okay. Your cells, interaction between cells. Your cells are loading in the matrix, supportive material. You have two chambers. You have two areas that you're going to find fluid. You're going to have in the body. You're going to have intracellular and extracellular. Intracellular and extracellular. Cytoplasm. About 67% of your fluid is intracellular. About 33% is extracellular, and of that 33%, 20% of that is your blood. The rest is in the tissue. Okay, extracellular, everything that's outside the cells. Everything passes through the extracellular fluid. You've got, your capillaries, oxygen, glucose, whatever, diffuses out of the capillaries into the into the cells that need it. But everything goes through at one point goes through the extracellular material. They receive their nourishment from and release waste into there. They communicate with each other by secreting regulators, i.e. hormones, i.e. neurotransmitters. Because you have nerves, you don't just have one nerve from here to here, though sometimes I think I do, and my employees get on it. You have synapses between the nerves. The nerves excrete extracellular material, neurotransmitters, acetylcholine. Don't write that down. We're going to get there. Anyway, the communication, hormones, extracellular, body fluids, 67% within the cells, 33% out of the cells. 20% of that 33 is blood plasma, and so about 80% is what they call interstitial fluid or the fluid in the tissues. Extracellular environment. Here's your epithelial. Here's glycoproteins in the extracellular matrix. We used to call them mucoproteins. Now they're glycoproteins. You have your epithelial membrane. Now, if this was the outside, if this was the skin, you'd have a layer of keratin on top of that. This is probably would be cuboidal or columnar epithelial, probably in the kidney because it's one layer. You get into the intestines, it's two layers. Yeah. There are seats up here. No, but you're right. I'm sorry. Okay. So, and then here's your blood capillary. So you have all your nutrients in that coming through the capillaries. Now look how they drew this. You see the spaces? The cells in the capillaries are not real tight. They're actually fairly porous. Not the cells, excuse me, the cell junctions. So that you want glucose and oxygen to be able to diffuse out of that capillary very easily. You want CO2, other waste products, to get into the capillary so it's carried off and taken away. Okay. Extracellular matrix. This is your extracellular matrix here. This is what's it makes up your extracellular matrix. Contains protein fibers, gel-like ground substance. Protein fibers have structural support. Collagen is one of them. They're in, it's in the matrix. Glycoproteins, proteoglycans, a type of polysaccharide. Integrins are glycoproteins that extend from the cell cytoskeleton and bind to the extracellular matrix. Let's see what they have. So an integrin might go from, say, inside this cell. I know it's in the cells in here, not necessarily an epithelium, but it will go through the cell wall. Protein goes through the cell wall, goes into the intracellular matrix, and attaches itself to the collagen or whatever, and it binds it. And that's a lot like what your basement membrane is made up of. It holds it in place. Integrins are glycoproteins that extend from the cell cytoskeleton and bind to the extracellular matrix. Cell transport. We talked about transport through the plasma membrane. It's selectively permeable. When we looked at it, you had bulk transport, you had phagocytosis, endocytosis, you release stuff through exocytosis. The plasma membrane is selectively permeable. It allows some things to cross. It does not allow proteins. It does not allow nucleic acids to cross. It doesn't want to lose them. It needs them to stay alive and reproduce. Generally not permeable to proteins, nucleic acids, or large molecules. Generally permeable to ions, nutrients, and wastes. I mean, obviously, the waste you want to get out. The cells have ion channels or ion gates, and we'll get to that. And nutrients. Obviously, you need to be able to get glucose and that into the cell, depending on the type of cell. A neuron uses a lactate rather than glucose. Now, we're going to talk quite a bit about the membrane transport. We're going to talk about how cells get substances across the membrane. You have carrier-mediated. You have the trucking company that's going to carry this stuff to the cell and get it in the cell. Or you have a non-carrier mediated. This is closed. The stuff's in the environment around you. You just simply absorb it. You have various channels that it can go through, non-specific aquaporins. You have ion channels that are just open. They go, the water can go back and forth. Not a lot of them. Not enough to cause problems, especially with the ions, because you're always pumping them. You're always trying to maintain a balance with that. Okay? This is, I mean, that's right out of your book. It's a table, it's right out of your book. Passive transport. Molecules move from a higher to lower concentration without using enzyme. Active transport, you're pushing them out. Generally, you're pushing them against a gradient, meaning that if you're taking them from a lower concentration and pushing them into a higher concentration. So you're creating a vastly different concentration between the inside and the outside of the cell. And it uses ATP. In active transport, you need energy. It uses a tremendous amount of energy to do this, especially in some places like the kidneys where you're reabsorbing. So, plasma membrane, nonpolar molecules. Plasma membrane is nonpolar. Nonpolar molecules generally just slide right through. You'll see that when we do the lab on Thursday. Here you have channel proteins. These are open. They're the aquaporins, the ion channels. They just let the stuff go back and forth through. And it moves through because of the gradient. And then you have carrier proteins. These are active channels, and they use, may or may not use energy at this point. So, we talked last week a little bit about osmosis. Diffusion and osmosis. I was going to bring something in, but that's okay. If you take a glass of water and you put a drop of food coloring in, it goes in nice and dark, whatever color you used, green. And then generally, eventually, it just kind of fuses, so the entire water bottle is, or glass is a light green, right?
Right.
Oh, thank you. I just wanted to see if I had put you to sleep already. Diffusion. I'm working on it. You know, last week I brought cookies. I should have brought no-dos. What's that?
That's a good word.
When you left?
That was not a good question.
I know.
What did you say last week? No. Diffusion. What? We put that drop of food coloring in, it hits the water, eventually it just moves through the water. Now this actually follows the second law of thermodynamics, because when it's nice and concentrated and green, it's more organized. You put it in a glass of water, it spreads throughout, it's less organized. That's just a bit of information. Anyway, you have the molecular movement, the thermodynamics of the molecule, the electrons spinning around. It causes what they call a Brownian movement. And it just, so the electrons are just kind of bouncing around there, and as long as the concentration's lower, where you didn't put the drop of food coloring in, this movement is going to move it out until it's equal. And then they still move back and forth, but you can't really tell because it's all the same color. It's not like they're all going to congregate in one area again. That is diffusion. That is diffusion. It diffuses. A solution. So we know now we're going to give, I don't know if I put that on there or not. Solvent and solution. Solvent. If I have a saline solution, the solvent is water. The solute is salt. Saline is salt and water. Salt. Okay? Solute is the hard stuff. Solvent is the liquid. Molecules in a solution are in a constant state of motion. That's the Brownian movement. If there's a different concentration between two regions, the random motion will establish an equilibrium via diffusion, just like the food coloring. Okay? What's happening here is, this is not a membrane, but we've got a higher concentration here, lower concentration here of whatever, little black balls. And through diffusion, it evens out. It just evens out. Here there's a net diffusion from this side to this side, and then when you hit the equilibrium, there is no net diffusion. They do go back and forth, but there's no concentrating on one side or the other. Net diffusion, due to random movement, the net direction of diffusion is from the high to the low solute concentration. It will happen across the permeable membrane. That one took me by surprise. So what happens is if I've got a membrane here and I have glucose in it, small diffusible molecules, they're going to go in and out. They're going to go in and out because they can, and they were more concentrated in here, so they're going to move out until it's equal. The protein will not. The protein will not because it's too big. It's too big to go through the pores. So if you have something that you have a higher concentration on one side of the membrane than the other, and the membrane is permeable to that, it will diffuse across until they're equal, or if in the case of a red cell or something like that, it crenates or bursts. Diffusion across the plasma membrane. Nonpolar, lipid-soluble, usually pass pretty easily. Oxygen, CO2, and steroid hormones go across too, generally because of their smaller size. Gas exchange. In the alveoli, net diffusion of O2 into the cells and CO2 out of the cells is because of the concentration gradient. Remember, in the Krebs cycle, you have water and CO2 at the end of the Krebs cycle. The cytoplasm of the cell fills with the water and CO2. When it hits a point where CO2 is less on the outside, it diffuses out. And it's used oxygen as the electron receptor, so it's turned all its oxygen into water, so there's more oxygen on the outside that diffuses back in. There's not an active pump. It just goes from an area of high concentration to an area of low concentration. And that's what happens in the alveoli. Okay, tissue cells. Oxygen, carbon dioxide. Oxygen's more prevalent here. It goes in. Carbon dioxide, more prevalent inside. It goes out. You're never going to get it all in or all out, but it's done us pretty well so far. Charged ions can pass through ion channels that cross the plasma membrane. The channels may be open or gated. That's what was in that first slide. Here's your ion channels. They can be open or they can be gated. Open, the ions just pass through. And why do they pass through? Because you have a higher concentration on one side than the other, and diffusion will push it through. It's called a concentration gradient. Higher on one side, higher outside than inside the cell for sodium, higher inside the cell than outside the cell for potassium. And we'll get to that. Okay, here's diffusion across the plasma membrane. Here the gate is open, or excuse me, the gate's closed. Here the gate is open. And you know, maybe a protein that binds, or hormone, or a nerve impulse that opens these gates. But here, your ions, sodium, if it's going in, it's probably sodium, are just going right through the gate. Here they can't. The gates are closed. Rates of diffusion. Now, on Thursday, we're going to take an egg. Each table is going to get an egg. And this egg has been put in vinegar to dissolve the shell. And then we put the egg in Caro syrup. Caro syrup is osmotically much more concentrated than the inside of the cell, so the fluid from the inside of the cell goes out and the eggs shrink. Then what we're going to do is put the eggs in distilled water, where you have a bigger concentration of solute inside the egg, and you're going to watch that water that has gone out of the egg go back in the egg. And you do it by weighing the egg. What happens here is the bigger the concentration difference, the faster that diffusion is going to occur. And it starts quick, and then as the concentrations become kind of equal, it levels off until they're equal. Temperature of the solution, magnitude of concentration, permeability. If the membrane is very permeable to those ions or that substrate, solute, it'll go through faster. Temperature of the solution. Temperature of the solution goes up, it's moving more. There's more movement to move it across. Surface area of the membrane. Obviously, if you're trying to put something into the size of a sugar cube, through the size of a sugar cube, it's going to go pretty slow, as opposed to a balloon or something like that. And then your kidneys and your intestines, you have what they call microvilli, and we talked about that. The folds lining the kidneys and the intestines. That just increases surface area. So if I have, if I didn't have microvilli, the inside of my intestines, might look like that, and all I have is this surface area to absorb. However, with the microvilli, if I go like this, you could, just by measuring the distance, so that see how much more surface area you would have. That's what the microvilli do. They just increase the surface area for a much more rapid diffusion across the membranes.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
Osmosis. Now this is kind of where the rubber meets the road here. Osmosis is diffusion across a semi-permeable membrane. Water molecules do not carry a charge. They can pass through the plasma membrane slowly. They can go through, or they can go a few of them can go right through the membrane. More of them go through the pores, the aquapores. Because of this diffusion, solute instead of solvent, the diffusion of solute, let's go back. Because of this, the diffusion of solute instead of solvent is unique. Generally, with osmosis, what's moving is the solvent, not the solute. Aided by channels in the membrane called aquaporins, water will get in some tissues. Requirements for osmosis. A solute concentration difference. Solute concentration difference. On either side of a membrane permeable to water. The membrane must be impermeable to the solute. If the membrane's permeable to the solute, it's just going to go across and diffuse wherever it wants to go, just like in that animation. The concentration, yeah, if it's not, it has to be impermeable, or the concentration difference wouldn't be maintained. Solutes that cannot cross in permanent osmosis are called osmotically active. So if I had, say, that lump of sugar again, but instead of just dropping it into a beaker of water, this beaker has a semi-permeable membrane. Well, if I take the sugar and put it in this side, you're going to have the concentration of the sugar, which is a solute, on this side, and then you've got nothing. You've just got water on this side. So by the rules of osmosis, the water is going to try to go across in here until there's no osmotic pressure or the concentrations are equal. Now this happens in the cells. The places where you have the cell membrane is semi-permeable, either extracellular or intracellular, you've got a higher concentration of an ion, of a substance, gas, of glucose, something like that. It's going to cross as long as it's in a semi-permeable membrane. Got that? Because we build on that, so let's make sure you got that. Osmosis is across the semi-permeable membrane. The solvent is going to move from an area of low solute or high solvent into an area of high solute or low solvent until they're equal.
But don't they say that membrane in the middle, it has to be impermeable to the solute, and the solute being...
Yes, impermeable, not permeable. The solute can't get through, so the only thing that can move is the water. Okay? Let's go. Here, this would be a model of osmosis. Let's see what we've got here. You have water, the small blue ones are able to move back and forth. It's more dilute here. This would be your solute. So it's more dilute here, and the solute is more concentrated here. It's going to draw water across until the ratio is the same. Okay?
The water is the solute.
The water is the solvent. Whatever this is, protein, sugar, is a solute. The solute can't move across. These pores are too small. The solvent is the only thing that can move across. Net movement of water from the side with more water, more dilute, to the side with less water. More water, less solute. Less water, more solute. It's going to try to even it out. It'll just move across the membrane. However, moves from low solute to high solute concentration. So I just went over.
Are there any tissues where osmosis and diffusion both occur?
Well, yeah. I mean, even in your cells, you would have diffusion because you have the pores, like the aquapore or the ion gates, that are open gates. That's simple diffusion.
Which one moves faster, the solute or the solvent?
Well, the solute's not supposed to move at all, except coming through the open gates. That's where your ions or your glucose or whatever would get in.
Diffusion moves.
I don't know which one would move fast. Again, the speed is in the concentration gradient. Osmosis. Here you have a semi-permeable membrane. You've got 360 grams per liter of sucrose. In the outside, you've got 180 grams of glucose. Grams per liter of glucose. Which way is the water going to go? The water is going to go in. It's going to go in until 40, what's 540 divided by 2? 250, 270. It's good. The water is going to go in until enough water has come out of here and gotten into there that they both have about 270 grams of glucose per liter. They both have the same concentration. Then it's just going to go back and forth.
But what, okay, I'm sorry, but the membrane will expand? How does it?
Yeah, well, okay. That's a good question. In this case, the membrane has to be able to expand to let the water in. When we do our lab on Thursday, we're going to use red blood cells. We're going to use red blood cells. They expand, but they break pretty easy. So you're going to see if something comes in like that in a red blood cell, it'll probably hemolyze it or burst it. If you put a red blood cell in, well, it's an answer to your questions. Yeah, so this, to have this actually work, it would have, the membrane would have to be able to expand. Now, when we do the egg, we put the egg in distilled water. I don't care how much water you can put in that egg, you're never going to get it to zero. Distilled water has zero solute. So the water is going to keep going into the egg. What stops it?
Membrane.
The membrane. Right. It can only, and the egg membrane's tough. So they swell up nice and, like water balloons. But and then they stop because the oncotic pressure isn't enough to rupture the membrane. In a red blood cell, the oncotic pressure, or the pressure of the water going in, is enough to rupture the membrane, and the cells hemolyze.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules, such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Okay, let's think back. I think I had the hyperiso, all that on there. Okay, let's think back for a second. Remember, when we were talking about the absorption of glucose in the cells, and glucose gets into the cells, i.e. the liver cells and that, it said you don't leave glucose as glucose molecules. Because of the osmotic pressure it would create. And it would suck water in until the cells die. So what happens in the liver? What does it do to the glucose? Changes it to what? Glycogen. So if I take one, say I took 100 molecules of glucose, and that would exert the oncotic pressure of 100 molecules. If I made it into one molecule of glycogen, that would exert the oncotic pressure of one molecule. Now this is kind of important when we start talking about the ionization in that. So if I take, it's just like what they did with the urea here. By the urea binding the water, it's taken what was 3 molecules and made it 1. So it cut the osmotic pressure on this side by 2/3. And the water's just going to come back through. Because there's more water on this, more free water on this side than there is on this side. So until it balances out, the water's going to move through the membrane that way. Now, again, if they had a couple of urea on this side, it could actually balance. But obviously, if there's no urea over here, it's going to try to draw all the water to this side. And that probably won't happen. But you will in some of the experiments, not any that we do. We used to do it with molasses and semi-permeable membranes, and it was just an ugly mess. So we went to the egg. Osmotic pressure. So what happens is when you have a high concentration of solute on one side of the membrane and you have a high concentration of solvent on the other side, the solvent's going to move across. The solute exerts osmotic pressure. The solute exerts osmotic pressure. It's going to draw the water across. Osmotic pressure is the force surrounding a cell required to stop osmosis. Or it's the force that's drawing it in. Can be used to describe the osmotic pull of a solution. The higher solute concentration would require a higher osmotic pressure. So, like I said, the more concentrated it is on one side, the more it's going to draw water through. It's exerting osmotic pressure on here for the water to come through. It's the gradient. It's the pressure. So here, let me see what they've got here. You have water, 100 grams per liter of sucrose. In there, the water is going to go in. You have 360, the water is going to go in. I'm not sure what they're trying to show you in that one.
Second one?
Here.
Oh, it's because like it can't push hard enough to go past.
Oh, I'm sorry. You're right. The membrane, like the egg, it's swollen as hard as it can, as big as it can. If they did, the solution was initially, okay, I didn't read it right, 180. Look at the size here. It's expanded as far as it can. So there's no more osmosis, no more water can come in. Same size, 360 still. The osmotic pressure is there, but nothing can happen because it can only absorb so much water. Okay, a mole of a compound. A mole of a compound is measured in its molecular weight in grams. The number of atoms in 1 mole is always the same no matter what the compound is. 6.02 times 10 to the 23. I believe that's Avogadro's number.
It is.
What? It is. You know how long it's been since I even said that word?
So we should worry about it.
No,
Oh, you have to worry about it.
I'm not gonna,
Just
unless when I'm writing the quiz, I remember.
Probably not.
Probably not. You can make molar solutions 1 molar or mole out. Solutions molar or mole out. Now let me tell you the difference. You have, if you take a beaker and you put 58.5 grams of salt in there, that's 1 mole of sodium, 1 mole of chloride. And you take that beaker and it's got a line of 1 liter. And you fill it so it comes right up to the 1 liter. That's, what's that? If I get it wrong, you'll let me know, right? That's a 1 molar solution. QS, quantity sufficient to 1 liter. So you throw all this solute in, you add enough water to bring it up to 1 liter. That's a molar solution. Now, if you're using copper, I don't know if it'd ever do it. Yeah, we'll use copper sulfate. Yeah, that's a lot heavier. So you've got a lot more in there. So what happens is the ratio between the different substances would be different. The ratio of water to the different substance. A 1 mole solution. So what we normally will use is molality, not molarity. Though molarity seems to roll off the tongue easier for me. Molality is 1 mole of solute plus 1 liter of water. Not quantity sufficient to plus 1 liter. Then you have a ratio between the solute and solvent that no matter what you use, the ratio is equal. So that's molar versus molal. That's not a bad test question.
What is that?
Now you're listening. Molar versus molal. Okay, glucose has a molecular weight of 180. To make 1 molar solution of glucose, molar solution of glucose, you dissolve 180 of glucose in water to make 1 liter of solution. Sodium chloride has a molecular weight of 58.5. To make a 1 molar solution, you dissolve 58.5 grams to make 1 liter of solution. Not useful for the discussion of osmosis, since the solute concentrations are different depending on the solute. More water is used to make a 1 molar solution of sodium chloride than it is to make a 1 molar solution of glucose. The ratios are different. That's why we don't use molar, we use molal. 1 molal solutions take the molecular weight and then is dissolved in 1 liter of water. The amount of water never changes, so you can compare the concentrations and predict the direction of osmosis. Molal. Okay, molality. I think we've beat this. You guys got that? Or you want 1 mole plus 1 liter. Mole of solute plus 1 liter. Okay? Osmolality. Now we're going to throw another curve in there. Is the total molality of a solution when you combine all the molecules within it. So, if I have technically osmosis, if I have a 1 mole solution on one side and I have a 1 mole solution on the other side, and the solute can't cross the membrane, the solvent just kind of diffuses back and forth. The Brownian movement, the thermal energy just moves it back and forth. Okay? But what this is saying is the total is when you combine all the molecules within it. So for instance, if I have sodium chloride on one side and I have glucose on the other side, and I have a 1 molar solution of sodium chloride, I have 58.5 grams plus a liter of water. On the other side, I have 180 grams of glucose plus a liter of water, 1 molar. You would think that it should be even, but it's not. Because on the sodium chloride side, you have 1 mole of sodium and 1 mole of chloride. That's the sum of all the atoms. So you have the osmolality is a total of the molality of a solution when you combine all the molecules within it. Glucose is 1 molecule. Sodium chloride is 2 molecules. So you're going to have an osmotic pressure. You have 1 mole here of glucose. You have 1 mole of sodium chloride. Here, your osmotic pressure is going to be 1. 1 molar osmotic pressure. Here, you're going to have 1 from the sodium and 1 from the chloride. 2 osmoles. The water is going to go that way.
It's not more constant.
It is. Osmotically, it's more concentrated. Molality, it's the same. The difference, molality is gram, is 1 atomic weight per liter. Osmolality is the number of ions. You take the molality and multiply it by the number of ions. So on this side, you just have glucose. On this side, you have sodium and you have chloride. So it exerts twice the osmotic pressure on this side, so it's going to bring the water over here. Okay? They're both. Now, if I wanted to make the solutions equal, I would have, like they say here, I would have to use either 360 grams of glucose, 2 molar, and a 1 molar, 2 mole, excuse me, and a 1 mole of sodium chloride, or else I would have to use 1 mole of glucose and half a mole of sodium chloride. Because sodium chloride exerts twice the amount of osmotic pressure. Okay? Okay, here it's saying the same thing. In this one, you've got 1 mole of glucose and 1 mole of fructose. So you've got glucose and fructose, so this is a 2 molar solution. And it exerts 2 osmoles because it doesn't ionize. Electrolytes that dissociate in water have to be assessed differently. Sodium chloride disassociates into sodium and chloride, and it has to be counted as separate molecules. That's what we just went over there. A 1 molar sodium chloride solution is actually 2 osmoles because it breaks down into sodium and breaks down into chloride. So what they're looking at here is the water.
The top one's flowing, going out.
The water's going out, yeah, until it equals. Until you come out, you got 1 mole glucose, 1 osmole, 1 mole of sodium chloride, 2 osmoles. So the water is going to go out until they both hit 1.5 osmoles. Then they're equal. Which is, just what we looked at here too. Once the osmolality is equal, there's no osmotic pressure. The water just diffuses back and forth. Plasma has the same osmolality as 0.3 molar glucose, or a 0.15 molar sodium chloride solution. These are considered isosmotic to plasma. Okay, tonicity is the effect of a solute concentration on the osmosis of water. Notice in that other video, that other action, it talked about being hypertonic, isotonic, hypotonic, tonicity. If I have there, I've got, tonicity is relative. It's not, you can't say a solution is hypertonic without telling you what it's hypertonic to. So for instance, right here, you have 2 osmoles on this side, 1 osmole on this side. This solution is hypertonic compared to the glucose solution. It's more concentrated. It's hypertonic. It's going to pull water in. The glucose solution is hypotonic, less concentrated than the sodium chloride. Isotonic is when they're the same. If a membrane separates 0.3 moles of glucose solution and a 0.15 mole of sodium chloride. So this is half the concentration of that because it dissociates into sodium and chloride. It's isotonic. There's no net water movement. Tonicity takes into account the membrane. If the solutes can cross, the tonicity changes. If you put an RBC and 0.3 molar of urea, the tonicity will not be isotonic. The inside of the cell is 0.3, but with urea can cross right into the red cells, and as they cross in, we're going to draw water with it, like you saw in that one slide. The cells will eventually burst. So if you put urea in red cells, which we'll do on Thursday, or you put something like hexane in red cells because it's nonpolar, it goes right through the membrane. It increases the osmotic pressure to bring water into the cell, and the cells burst. Solutions with a lower concentration than the cell are hypoosmotic and hypotonic. They'll pull water into the cell until, sometimes you get lucky, you get a young red cell and it's got either a strong membrane or the solution is close enough that it actually dilutes it out so they're equal, but usually the cell lyses or breaks up. Solutions with a higher solute concentration are hypertonic. They're going to pull water out of the cell. They're hypertonic compared to the cell. Again, tonicity, you have to compare it to something. You just can't say this is hypertonic. It's got to be hypertonic compared to something. It'll pull the water out of the cell and the cell will crenate. Here's a normal cell. In a hypotonic, the water is going to go in until the cell lyses. In a hypertonic, the concentration of solute is greater outside the cell, so the cell is going to draw water out of the cell to try to dilute it and make it equal. And the cell will crenate or shrivel up.
The image on the left is human erythrocytes, or red blood cells. The image on the right is enhanced with filters. These living cells are in isotonic solution. Note the lozenge shape. Now the solution has been changed to have a high salt concentration. It is hypertonic. The red blood cells shrivel and become crenated. Finally, the solution has been changed to plain water. The cells swell and burst, leaving empty membranes behind. This phenomenon is called hemolysis.
Questions? Okay. Regulation of blood osmolality. It has to be regulated. Because you have ions and that need to be pulled in and out of the nerve cells especially. And when we talked about the action potential of a nerve cell, you've got all this sodium on the outside and potassium on the inside. If you change the osmolality, either dehydrate it or overhydrate it, then that gradient is going to change and your nerves aren't going to fire right. And we'll get into that. Constant osmolality must be maintained. There's osmoreceptors in the hypothalamus, in the brain. They detect increases. Excuse me.
Gotta go.
Yeah, not for that one. Osmoreceptors in the hypothalamus detect the increase in osmolality, meaning that as your osmolality increases, your blood volume goes down, and it detects that. It triggers thirst. It triggers the secretion of ADH, antidiuretic hormone, which causes more water to be reabsorbed in the kidneys, and you urinate less. With a lower plasma osmolality, osmoreceptors are not stimulated, so more water is excreted in the urine. So you have dehydration, you have decreased blood volume, you have increased plasma osmolality, osmoreceptors, and this is actually a physical receptor. I mean, it's like a membrane that moves back and forth. Osmoreceptors in the hypothalamus increase ADH secretion, cause you to be thirsty, you drink more water, your kidneys take more water in, you intake more water, and you retain more water. You reabsorb more water. Carrier-mediated transport. Okay. Some molecules are too large to diffuse across the membrane. So your cell needs them. You're going to have to get them across. Or the cell wants to maintain certain concentrations of it between the inside and the outside of the cell. Carrier proteins within the plasma membrane move the molecules across. These proteins are specific. It's not a generic carrier protein that just takes everything in. They're specific. Sometimes the proteins can transport more than one, so I guess they're not that specific, but they're pretty specific. But then there's competition. For instance, if you have a protein that will take sodium and glucose into the cell at the same time, and you've got sodium and glucose in the extracellular fluid, they're going to compete for binding sites on that protein to get into the cell. So each one is going to go in at a fraction of the rate as if it were not competing. Transport rates increase with increased molecular concentration until saturation is met. The proteins will transport, it's kind of like enzymes. The enzymes will, it changes it and then lets it go and goes to the next one. And the rate increases until all your enzymes are being used. And then the rate of increase stops and it levels out. The same goes on with this. The transport rates for getting the stuff across the membrane increase as long as there's free sites to transport it. When all those sites are full, then the amount and the rate of transport just hits the maximum and levels off. So if you have two molecules, the transport mechanism's saturated somewhere around here because both X and Y are trying to get in through that gate. If you have one molecule, then it's only one and it doubles because those gates are only trying to get one molecule. So when you hit the rate of it leveling off, it's higher for one molecule as opposed to having the competition for two molecules. Facilitated diffusion. Powered by random movement of molecules, no ATP is used. Goes from high to low concentration and requires specific carrier-mediated protein. That's why it's facilitated. Outside the cell, you have a higher concentration of glucose. You have a carrier protein. It comes in, it binds here, the proteins change shape, and it dumps the glucose in. That is not an active transport, but it is facilitated because they've got to go through these protein gates.
You should go back to the slide before this, please.
Okay, what no ATP is used. That's kind of important. So you can get glucose into the cell. You're not having to pump it in. It goes through the gates. Net movement is from the high to low concentration. The only reason the glucose is going to go into the cell is there's less in the cell than there is in the extracellular fluid. And it requires the mediated proteins, the carrier-mediated proteins. So here's the protein that allows glucose into the cell. Again, this is just diffusion. It's going from a higher concentration of solute. It's diffusion of the solute because it's going through a permeable membrane. But it's going from a higher concentration of glucose into an area of lower concentration. And it happens throughout all your cells, throughout the body. Facilitated diffusion. Transport proteins may exist in the plasma membrane or be inserted when needed. Some proteins, it's interesting that if, say for instance, there's plenty of glucose in this cell, in some cells, these proteins come out of the membrane and go back into the cytoplasm and just float around until they're needed again. Then they're inserted into the plasma membrane, and the glucose comes in until the concentration goes up. Then they're pulled back out. Muscle cells do this during exercise to transport glucose. We will get to that when we get into the muscles. Facilitated diffusion here. This is where the vesicle moves. When it's stimulated, it hits the plasma membrane, and it's there to bring stuff into the cell. Just what I was telling you.
So it's just diffusion with basically with help, basically?
Yeah, through specific channels, because the molecules are too big to go right across the membrane. They're too big to get, so there's got to be channels for them to diffuse through.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor, which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
Okay? The difference between facilitated is it has to have a channel. These molecules are too big to go through the membrane. So there's got to be a place for them to go through. they still move from an area of high concentration into an area of low concentration. That's the concentration gradient. You know, if you ski, you come from a high altitude to a low altitude. Active transport. Sometimes molecules must move from an area of low concentration into an area of high concentration. The thing about active transport is it uses energy. And these are protein pumps. Carrier-mediated proteins are called pumps. Now, you know, in the old days when I started in the lab, we operated on people's stomachs all the time for ulcers. Something that we almost never do anymore. Prilosec, they're a protein pump inhibitor. They go into the stomach. They stop the protein pump that pumps hydrochloric acid in, and you don't have the ulcers. The other thing that they discovered is 80% of the ulcers are caused by a bacteria called Helicobacter. And antibiotics take care of that. So you do very little gastric surgery like that anymore. Okay, molecules move from an area of low concentration to an area of high. It requires energy. It's done by the pumps. Occurs with the hydrolysis of ATP to ADP, which is directly responsible for the protein. The transport protein is an ATPase enzyme. The pump is activated by the phosphorylation of taking the phosphorus off the ATP, making an ADP. Carrier proteins, calcium. This is calcium that's going to come in. Here, in the cytoplasm, you've got a low calcium. In the fluid around it, you've got a high calcium. The body, the cells are actively pumping the calcium out. It uses ATP, the calcium comes in, ATP causes the protein to change shape, and boom, your calcium goes out. The calcium pump, located in all cells in the endoplasmic reticulum. Remember, in the endoplasmic reticulum of the muscle cells, it stores calcium. They store calcium to help with the muscle contractions. Removes calcium from the cytoplasm by pumping it into the extracellular space. It creates a strong concentration gradient for movement of calcium back into the cell. Aids in the release of neurotransmitters in neurons and muscle contraction. That's part of the calcium there that aids in the muscle. Your body is pumping it out, getting a high concentration difference between the outside and the inside of the cell. So when you need that calcium rushing in to do the contraction, there it is. It also aids as neurotransmitters. When you hit the synapse, you have neurotransmitters that are released to carry that action potential across the synapse and down the next nerve. Sodium-potassium pump. Again, found in all body cells. It uses ATPase again. It's an energy-using function. You pump 3 sodium out of the cell for each 2 potassium you bring in. It serves 3 functions. It provides energy for coupled transport. And we'll talk about that in a minute. It produces electrochemical impulses in the neuron and muscle cells. Your depolarization of a nerve, your action potential, it's not really electrical, it's really chemical because of the movement of sodium and potassium through the cell walls. These are gates that open up to allow these chemicals, the high concentration of sodium outside the cell to go in, and the high concentration of potassium inside the cell to go out. And we will get into that. Maintains osmolality. 2 potassium in, 3 sodium out. 2 potassium in, 3 sodium out. And it uses ATP. That's all the cells have.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Okay. Your sodium potassium pumps. Your active pumps require energy to move ions or whatever from the low concentration into the high concentration. Exactly the opposite of what would happen with diffusion. Secondary active, called coupled transport. Sometimes when the sodium comes in or another ion comes in, it grabs another molecule and brings it back in with it. They found that sodium does this with glucose, but the energy, it's called coupled transport, the energy to move the molecules across by their concentration gradient is acquired by moving sodium back into the cell. So you've got a lot of glucose or something outside the cell. You've pumped all this sodium out. So you have this huge concentration of sodium outside. When it comes back in, and it does, it leaks back in, that's why you're constantly pumping it out, it'll bring these other molecules in. Since sodium was originally pumped out of the cell using ATP, this is considered active transport. So you pump the water back above the dam, and then when it comes back through, it carries another molecule with it. Co-transport. The other molecule is moved with sodium. Coupled transport. And I think coupled transport and co-transport are really pretty much the same thing. The molecule is moved with the sodium. It's one of the common ways to get glucose in. Counter-transport is if you're bringing the sodium in, and it's kind of like a teeter-totter. You're throwing the other one out when you're bringing the sodium in. But it's a physical. It's not a chemical. It's a physical. When that sodium ion comes in, it hauls the other one in with it. Extracellular fluid, you have the sodium and the glucose. The concentration of sodium is on the outside because you've pumped it out. Your sodium potassium pumps have pumped it out. Now this sodium concentration and the glucose, they're attaching here by their conformational change. The protein opens up. The sodium moves down its concentration gradient because you're going from high concentration out here to a low concentration here. Now it's hauled this glucose in with it. And the glucose may be more concentrated in the cell, but the sodium's just bringing more in. This is coupled transport. The sodium and the glucose are coupled to get in. Okay? Questions?
Small molecules, such as sugars and amino acids, can be transported up a concentration gradient. The sugar moves via a membrane transport protein from outside of the cell where the sugar concentration is low to the inside of the cell where the sugar concentration is high. The transport of the sugar through a coupled transport protein is driven by the movement of counterions, such as sodium ions or protons, that are moving down their concentration gradients from a region of high to low concentration. Sodium ions and the specific sugar or amino acid simultaneously bind to the same transmembrane protein on the outside of the cell, called a symport. When the counterion is sodium, the low concentration of sodium on the inside of the cell required to transport the sugar is maintained by the sodium-potassium pump, which is powered by ATP. In a related process called countertransport, the inward movement of sodium ions is coupled with the outer movement of another substance, such as calcium ions. As in cotransport, the sodium ions and the other substance bind to the same transport protein, called the antiport, but in this case they bind on opposite sides of the membrane and are moved in opposite directions. The low internal sodium ion concentration is maintained by the sodium-potassium pump.
Symport and antiport. Both come in, or it's antagonistic. One goes out and one comes in. But you see, it's just constant. It's one of the ways the body can get the sodium out, but then still bring in or also get rid of things that it wants to get out of the cell. Yeah.
So when the potassium comes in, it also requires ATP energy? Because I know when the sodium goes out, because it's a higher concentration outside.
When the potassium comes in, it doesn't require any additional ATP. The pump uses a 1-phosphorus, that one bond, to throw 3 sodium out and bring 1 potassium.
So
it is active transport. It's still considered active transport.
Is the potassium inside a higher?
Oh yeah.
Higher.
Oh yeah. Your potassium, in the cell is about 30 times more concentrated than the potassium in the extracellular fluid. Whereas in potassium and sodium, we use milliequivalents per liter, which is a mole times the valence. But potassium inside the cell, it will be somewhere around 120, 140. Potassium outside the cell will be about 4, between 4 and 5.
And then so the co-transport is just from lower to higher, that's why it doesn't need.
Well, the co-transport is not, it's the result of active transport. But with the co-transport here, we're not using any additional ATP. We're simply using the concentration gradient of the sodium, or in other cases, the concentration gradient of hydrogen. So, because you do have hydrogen pumps that pump hydrogen out and bring sodium, I think, at the same time. We'll look at that. But this is just the sodium coming back in, hauls the glucose in with it, or sends the calcium out. But it's considered active transport because to get the sodium out, you had to use energy. Did that answer your question? Okay.
I have a question. The co-transport and the coupled transport, transport, they're different, right?
No,
Not
they're
the
pretty
same.
much the same thing.
Are they?
Yeah.
Because I thought the coupled was when they're coming down together, and the co-transport when they're the opposite direction.
No. Co-transport, symport is when they're both coming in, and antiport is when we're kicking one out.
Okay.
But here they call in the book, they call it antiport countertransport.
Yeah, the same antiport is the same as countertransport. This is co-transport, though. We're talking about co-transport. Counter means they're going in opposite directions. transport across the epithelial membranes. In your kidney, in your gut, you're pulling, in your gut, you're absorbing. You're eating the food. The gut is actually considered outside of the body, but you're absorbing the glucose and the proteins and all that from the gut and you're getting it through the columnar epithelial cells, in through the intercellular matrix, into the bloodstream, to the liver, or to wherever it's going to go. Involves transcellular transport, movement of molecules through the cytoplasm of the epithelial cells. So your epithelial cells, some of them, let me see if we get into that. You have transcellular, you have paracellular, where it moves between the gaps between the epithelium. Transcellular might be like in the brain, where your junctional complexes are so tight that nothing's going to move in unless it goes through the cell. Paracellular transport is limited by the junctional complexes. Remember, the junctional complexes are what holds the epithelial tissue together. You have the tight junctions, where the cell's walls are essentially fused together. Nothing's going to get through there. You have adherent junctions, where the cell walls are close together and they've got almost like a stitching between the two. And then you have desmosomes, where it's just like you tack them together, if you were to use a sewing term. You just put a stitch here and a stitch there just to hold them together. Did I get that right?
Just button.
What do you know about this? No, I'm just kidding. Okay, tight junction. Tight, right up there. Adherent junction, just kind of, stitched together. And desmosomes, they're just, kind of tacked, just enough to hold them together. See how wide it is? See how you got more space in there? And here they're just tight. Here you've got space. Here you don't. You don't get stuff through there. That's where you have to get that product, the glucose, the protein, whatever. And it goes in and it's absorbed through the cell membrane of the epithelial cell, goes transcellular, moves to the other side of the cell membrane, and is through exocytosis or through a pump, it's pushed out into the intracellular matrix and into the bloodstream to go where it needs to go. And many different types of carrier proteins at both ends of the epithelial. Movement of glucose across the intestinal wall is an example. Like I said, you get all this glucose in your gut from eating, and it's got to get into the bloodstream. It doesn't go between, it gets absorbed by the epithelial cells, and then goes out, and it goes out the other side and is excreted to where it can get in the bloodstream. You have blood. Here you have your lumen of the kidney tube. Okay, this is kidney. Here's your junctional complexes. What happens is you've got all these folds to make more surface areas. What's that? And the lumen of the kidney or small intestine. Sodium's higher concentration, glucose is lower. Co-transport is going to bring the glucose in with the sodium. So you get the glucose in, it moves through the epithelial cell, and it's transported out, and it's into the blood to go where it needs to go. And this is active transport. The kidneys use a tremendous amount of energy to do this. Transfers sodium down its concentration gradient, provides energy for glucose, and this is your cotransport to be moved against its concentration gradient. So you're pushing the glucose into the blood. ATP is used to move both the sodium and potassium against their concentration gradients. So that's the sodium-potassium pump that we just saw. It'll pump something out to reabsorb something else. It happens in the kidney, it happens in the intestine. Bulk transport. Large molecules. So have you got that? The smaller stuff. It's absorbed by the epithelial cells, or some of it, once it gets in there, can move through the junctions of the epithelial cells. But then it's pushed to the opposite side, out into the cellular matrix, and where it can be absorbed into the bloodstream and transported around the body. Now here, sometimes you've got large molecules, proteins, hormones, neurotransmitters. They're secreted via exocytosis. Now if you go back, remember in chapter 5, you have the nucleus. You know, it's got to do its thing, which is part of which phase? Do I dare ask that? That test is long behind you. Interphase. The cell's just doing whatever the cell was made to do. You know, the hormone-producing cell is producing the hormone, or whatever. So what happens is the cell is making whatever it's supposed to make, large molecules, and then they go into the Golgi apparatus, which is the mailroom. They're packaged in vesicles, and they're secreted via exocytosis. You know, instead of the membrane going around and bringing something in, it actually goes back this way and pushes it out. And this happens pretty quick. You look at your action potential and your acetylcholine or norepinephrine, they get out pretty quick because these action potentials are very, very fast. It involves the fusion of a vesicle with the plasma membrane. We talked about exocytosis. Requires ATP. Molecules with cholesterol into the cells requires endocytosis. And that's where the cell membrane invaginates. The molecule comes in, it seals, and you've got the vacuole. And it's either digested or, you know, becomes part of the lysosome, but it's used within the cell. Transport proteins interact with plasma membranes to trigger it. So they've got a receptor. They've got a receptor. It's most of the time it's receptor mediated. Invagination, they forms a pouch, it forms a vesicle. There's endocytosis. Exocytosis, you've got the vesicle, it combines with the membrane, it opens up, and whatever's being excreted goes out. And in the case of nerve cells, it happens in milliseconds.
It also requires ATP.
Actually, I think both of them require ATP, actually.
It was specified on your notes for the exo, but not for the endo.
I'll have to check that.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use three basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
Okay. Why don't we stop right here? I want to take a break. Go to the cafeteria and get something to drink or whatever. Let's come back. Let's start again in 20 minutes, 8 o'clock.